Hi, this is a quick video about the scientific method and how we're going to use the scientific method this semester in this course. So just starting real quickly, you've probably all seen this general diagram of the scientific method before, and depending on which website you look at, this kind of varies a little bit, but basically, this is a method that all scientists use for carrying out their research. And we're going to go through these different steps today and talk a little bit about what they mean and how we're going to use these steps to carry out experiments this semester in this course. So just real quickly, the basics of what the scientific method is. It's a systematic way of conducting scientific experiments. And as a scientist, I find this very reassuring to know that all scientists, whether they're in psychology, cell biology, physics, chemistry, everyone is conducting research in a uniform manner, following a uniform established set of steps. These steps are the same for all scientific studies, regardless of the discipline. And the purpose is to help scientists find cause and effect relationships or relationships between different factors that we can make conclusions about and use that information to build on previous research, to make health decisions, to develop new cures for diseases, to explain natural phenomena happening in our world, all kinds of reasons for doing this research. This is the uniform set of steps that all scientists follow. And it basically starts with the scientist asking a question and then designing an experiment to answer that question. And it's done in a very precise way so that when ultimately data is collected and analyzed and conclusions are made, we can trust that that information is valid because that scientist followed a uniform set of steps. So just thinking about why we should use the scientific method for doing research, why it might be important to have that consistency in how we can conduct studies, I think it's really, really important to think about the opposite, and that is information we read on the internet or on social media. And especially during the whole COVID era, all the misinformation that was out there that really had no scientific basis because it wasn't coming from a reputable scientific journal, a published scientific study, using the steps of the scientific method to conduct that research. It was a lot of opinions. And being able to discern what is opinion versus what is based on scientific data, that is a very important distinction in our world today. So the scientific method has probably never been more important to understand than in this era of misinformation. Really, when a scientist asks a question and they need to design an experiment, they're going to do some background research on what other scientists have done before them. That's really important because we're either building on previous research or we're answering a completely new question, but someone along the line has probably done something related to our research. And we want to find out everything that has already been done so that ultimately, when we construct what's called our hypothesis, we're doing that based on knowledge, not just a wild guess about what might be happening. That hypothesis is extremely important because we're going to design an experiment and collect data based on that hypothesis. So, what is a hypothesis? A hypothesis is a research question that includes the predicted results. So it's not just an open-ended question for debate, it's already predicting the relationship between two or more variables, and it really is going to be the basis for our experimental design. So in this course this semester, we are often going to do labs in which you're going to write a hypothesis that is going to be your prediction of the relationship between the factors we're studying. So for example, the effects of pH change on enzyme activity. You're going to make a hypothesis about that and design an experiment and test that hypothesis in this class. I don't know why I'm having trouble saying hypothesis right now, but I mentioned a research question a second ago. And a research question is really open-ended, whereas again, a hypothesis is going to go ahead and predict the relationship. So that's the difference between a research question and a hypothesis. A hypothesis is a very precise and succinct wording of that prediction. It doesn't ramble for multiple sentences or paragraphs. So it's really important that when you write your hypothesis, you do it in a very concise way. It has to be something that's testable, and it has to predict the relationship and the outcome. So for example, these would be two hypotheses in science. Basal plants that receive fertilizer have faster growth rates than unfertilized plants. So it's looking at the relationship between fertilizer and growth rate, and it's specifically in basal plants. And it's predicting the results. It's saying that the fertilized plants are going to have a faster growth rate than the unfertilized plants. Here's another one. Men over age 60, that's a very specific group of people, who exercise at least 30 minutes per day have a lower heart attack rate than those who exercise less than 30 minutes a day. So it's really looking at the effects of exercise on rate of heart attacks. And it's in a specific group of people, and it's really defining what that exercise means. So this is a very specific question. The researchers would then design an experiment to test this hypothesis, and this would be the whole basis for their research. It's also really, really important to note that in science, a hypothesis and a theory are two very, very different things. So a theory in science is not a predicted result. So we use the word theory in everyday speech in a very different way. Okay, so we can say, I have a theory about why my car isn't starting this morning. That's really a misuse of that term. What we're really saying is I have a hypothesis about why my car isn't starting this morning. A theory is not a prediction. In science, a theory is something that has been tested over and over and over again, and the same results have been obtained. So there aren't that many theories in science. In biology in particular, there's the cell theory that living organisms are made of cells. We pretty much have established that that's true. That's been proven over and over and over again. The theory of natural selection, which we'll talk about this semester, that is a big overarching theme in biology, and it's something that's been supported by considerable volume of data. So considerable volume of research and data that have consistently arrived at the same results. Only then can something be called a theory. So you can't say I have a theory about the relationship between fertilizer and basal plants. That's a misuse of that term. That would really be a hypothesis. Okay, ultimately, going back to our steps of the scientific method, after we construct a hypothesis, we need to test that hypothesis with an experiment. And experimental design in science is hugely important. If you start really reviewing scientific literature, you can start to poke holes sometimes in experiments, looking at things such as, were there other factors that could have been influencing the outcome? Were they really just testing that relationship? Was there something about their test subjects, such as the number of test subjects in the study, that puts the results in question? So experimental results are only judged based on two things. One is statistical analysis of the data, and the second is how solid was their experimental design. And this semester, you might already have experimental design that is pre-existing. So in other words, you have a lab procedure that's already laid out for you, or you might sometimes be asked to design your own experiment. And it's important to understand the things you should consider in designing this experiment. Whether that procedure is already outlined for you or not, there are some factors you're going to have to name in your lab reports. So let's go through what those are and what we should be considering when designing an experiment. So in an experimental design, there are some important terms that you're going to have to know. What is your independent variable in the study? What is your dependent variable? What are the controlled factors? What is your control group, which is different than a controlled factor, versus your experimental group? And then within that experimental group, what different levels of treatment are you studying? So let's talk about the definition of those terms, because again, you're going to have to identify all of those things in the experiments that you carry out this semester. So let's talk about what these terms mean by looking at a hypothesis that we previously mentioned when we were defining hypothesis. So the effects of fertilizer on basal plant growth. This is what we're studying. And in every experiment, there is what's called the independent variable. And this is the factor we are manipulating or changing in the experiment. So in this experiment, we are manipulating or changing fertilizer. We're giving basal plants probably differing amounts of fertilizer, and we're seeing how that affects basal plant growth. So in this experiment, our independent variable is the amount of fertilizer. Something that's very, very, very important. In every experiment, there can only ever be one independent variable. In other words, if we were changing the amount of fertilizer and the amount of water that a basal plant was receiving, we wouldn't know what was causing that plant growth. We wouldn't know if it was really the fertilizer that was affecting it or if it was the amount of water. Then what if we change the amount of sunlight too? The more factors that differ in this experiment, the less likely you are to make the conclusion that it was based on the fertilizer. So we really want to make sure the only thing we're manipulating or changing in this experiment is the amount of fertilizer. So every experiment can only have one independent variable. And that's very, very, very important. Okay, then what is the dependent variable? Well, let's think of it this way. In this experiment, we're saying that plant growth is dependent upon the amount of fertilizer we're applying. So in this case, the dependent variable is going to be basal plant growth. In other words, it's the factor, oops, sorry. It's the factor that is affected or changed due to the independent variable. So the independent variable is fertilizer and the amount of fertilizer. We're saying the amount of fertilizer applied is going to affect basal plant growth. In other words, basal plant growth is dependent upon the amount of fertilizer. The more and more examples of this you see, the more that will make sense to you. Controlled variables are also incredibly important in experimental design because, as I mentioned, there can only be one independent variable. And since there can only be one independent variable, that means everything else in the experiment needs to be kept the same. So these are the things we're going to control. And that means all the basal plants in this experiment have to be completely treated the same except for the amount of fertilizer. So let's think about some things we would need to keep the same in a study such as this. And I've already mentioned a couple of those. So this would be things such as the amount of water that the plants receive, the amount of sunlight that they're exposed to every day. What would be some other things that we should try to keep the same? How about the type or species or hybrid of basil plant? We might want to keep that the same. Maybe the age of the plants, because maybe older basil plants have a different growth rate inherently than younger plants. So we would probably want to keep that the same. We'd want the general location of those plants to be the same. The temperature. These are all things that you want to control or keep the same for every single plant in this experiment, so that when we see that growth rate, we can know that the only thing that differed was the amount of fertilizer. And we can therefore make conclusions on how the fertilizer affected the plant growth. So these are sometimes harder to identify in an experiment, but these are really important things to keep in mind this semester. And you will have to identify the controlled variables for most of our experiments. A couple of other terms. The control group is something very different from the controlled factors. The control group is the group. Sorry, I'm not typing very well today. I'm used to writing this on my iPad. The group that receives no treatment. What do I mean by no treatment? Well, in this case, our control group would be the group that receives no fertilizer. So, e.g., the plants that receive 0 fertilizer. That's our control group. We need a group that doesn't receive fertilizer to compare to. Okay, otherwise, we don't know what the plant growth would have been without fertilizer. How do we know fertilizer even affected plant growth if we don't have a basis for comparison? So your control group is always going to be what you compare the experimental group to. Sorry, this should be a lowercase e. That just means for example. So the plants that receive 0 fertilizer, that's our control group. They didn't receive the treatment. The treatment is fertilizer. So our experimental group receives the treatment. So in this case, this would be plants receiving fertilizer. That's our experimental group. Different levels of treatment. Well, do we really want to give all the basil plants the exact same amount of fertilizer? Or maybe we want to see if more fertilizer makes a difference. So maybe we want to test plants that get 2 grams of fertilizer per day, and another group that gets 4 grams of fertilizer per day, and then maybe another one that gets 6 grams of fertilizer per day. Because if we just gave 2 grams a day, maybe that's not enough and we wouldn't see an effect. If we're doing the experiment, maybe we want to do different levels of treatment. And then obviously, you want to have a lot of replication to really make these results valid. So you want to have a lot of test subjects. So you would want to have a lot of plants in the control group and in the experimental group. And you would want a pretty even number in each group. You would want to randomly assign each plant what group they're in, but then you would also want to replicate the entire experiment. All of these things in your experimental design are going to give your study more validity. Collecting data is obviously a very important part of all of this. So now that we've designed our experiment, we're going to start conducting our research. And at some point, we're going to have to decide, is our procedure working or not? And maybe we troubleshoot it and we go back and we redesign our experiment, and then we come back and do it again. But ultimately, we're going to collect data and we're going to have to analyze that data and draw conclusions. So let's look at another pretend study and look at some data. There is going to be a separate video on tables and graphs because you will often have to graph your data in this class, but I will talk about graphs just a little bit today. So let's look at a pretend study. So let's pretend that we've noticed that our dogs seem to be gaining weight since we started feeding them bacon. So we have the question, is bacon unhealthy for dogs? Well, unhealthy. What does that mean? To really conduct an experiment, we would have to really decide what we want as our measurement of healthy versus unhealthy. So let's pretend that in our experiment, we've decided that we're going to look at how bacon consumption affects total blood cholesterol levels in dogs. And we're going to use that as our measurement of healthy versus unhealthy. So my specific question is going to be, does bacon consumption cause an increase in total blood cholesterol levels in dogs? Is that a hypothesis? No, it is not a hypothesis because it's not predicting the results. That's an open-ended question. So that would be a research question. My hypothesis is going to be something like this. Daily bacon consumption significantly increases total blood cholesterol levels in dogs. So that's the hypothesis I'm going to use to design my experiment. So now let's start identifying all of these things that we talked about before, the independent and dependent variables, the controlled variables. What is my control group versus my experimental group? And are we going to have different levels of treatment? Okay, here we go. Independent variable. So we're studying the effects of bacon consumption on total blood cholesterol levels in dogs. So what is the factor that we're manipulating or changing in this experiment? Okay, we aren't directly manipulating or changing the total blood cholesterol level. That's going to be the effects of bacon consumption, right? So what we're manipulating or changing is bacon consumption. Another way of putting this would be the amount of bacon consumed. And of course, by dogs. And the dependent variable, we're saying total blood cholesterol levels are dependent upon how much bacon was consumed. So the dependent variable in this study would be total blood cholesterol levels. Controlled variables. What are the things that we would want to keep the same amongst all of our dogs so we could say that that change in total blood cholesterol level was only due to the bacon consumption? We know that these types of studies are very hard to conduct in humans, right? Because you can't put humans in a lab and you're only feeding them what you want them to eat all day, every day. So these types of studies are much harder in humans. But if you have a dog, you could probably conduct this experiment with your own dog. To do this with a lot of dogs would be pretty tough. But let's think about the things that we would want to keep the same. How about the breed of dog? Definitely probably the age of the dogs, because maybe younger dogs wouldn't have a change in total blood cholesterol level, but maybe older dogs would. What about other food eaten? We would want to keep that the same for all the dogs. So everybody's on the exact same diet, eating the same quantity of food, the same type of food every day. The only thing that they're eating differently is some are eating bacon and some aren't. How about the amount of exercise that the dogs are getting? We'd probably want to keep that the same. So we could go on and on and on with these examples, but you really want to make sure everything you can possibly control is being controlled. And the only thing that differs amongst these dogs is the amount of bacon that they're eating. Again, pretty hard to do with living organisms. Okay, what is our control group? Again, this is the group that receives no treatment. And in this case, the treatment is bacon consumption. So our control group would be dogs that receive no bacon. These are the sad dogs in the group. And that obviously means our experimental group, these are dogs that do receive bacon. And then we might want to have different levels of treatment. In other words, different amounts of bacon. So we might have some that get 2 grams per day, some that get 4 grams per day, some that get 6 per day, etc. And again, you would want lots of dogs in each group, similar numbers in each group. Dogs would get randomly assigned to a group, and you would want to replicate this experiment. Sorry about my throat all of a sudden. So these are the different things we need to consider in designing our experiment. So let's say we've done all of this and we collect our data. One thing that you'll be doing this semester that's very important is you'll be averaging your data. So if you were looking at a data table that had thousands of pieces of data, it would be a lot harder to see those final numbers. But when we average the data is when we really start seeing these connections. That's why we always want to replicate experiments, because we can't have just one result be the basis for making our conclusion. A lot of times in this class, we'll average the data from multiple groups. So in other words, our replication will be that each lab group is doing the exact same experiment, and we're going to average that data. And we'll be graphing the average almost always in this class. You'll never be graphing usually your individual group's data. Okay, just looking at this data, this doesn't really tell us everything we need to know, but it's important to note this is called Table 1. So then we could reference this by number in discussion of our results. And it's got a title, The Effects of Bacon Consumption on Total Blood Cholesterol Levels in Dogs. So somebody could look at this and they would know what data we're showing here. Each column has not only a title, but units, milligrams per deciliter of blood. That's important as well. You always want to make sure you're including the units so that the reader knows exactly what data they're looking at. Bacon consumed per day, this was in grams. And it's not really the starting blood cholesterol or the ending that's important. It's the change in blood cholesterol level that we're looking at. Just looking at the ending blood cholesterol, that doesn't really tell us anything. We need to know what they started at and what they ended at, what the difference is. So just looking at these numbers, you can see that it does go up as bacon consumption goes up. So you can see we had a control group that got 0. And then we had 3 levels of treatment, 5, 10, and 20 grams of bacon consumed per day. But what really tells the story of our experiment is the graph. This graph is a picture of our results. And it's very, very important to include a lot of things in your graph. One is that it needs to have a title. In this case, figure 1 goes with table 1. So that when we discuss this in our results, we can have the graph number be the same as the table number. So the reader knows this is a graph of that table's data. Needs to have same title. X and Y axes have to have not only a label, but units. And we also want to make sure that each axis has even intervals so the horizontal axis is our X. By the way, the horizontal axis, the X, that's always your independent variable. So in this case, that's the amount of bacon consumed. And then the Y, which is our vertical axis, that always needs to be our dependent variable. Oops, blood is misspelled here. But you can see that they're also not just a label, but units. You always want to make sure those units that were in your table are also on your graph. And again, even intervals, so we can look at the relative amount of change. And with this, you can automatically see, oh gosh, as the amount of bacon consumed per day went up, the change in total blood cholesterol went up as well. And you can see there's a legend over here that tells you what that line means. And then, of course, we can look at this visually and see that it increases, but is that change really significant? We only know that if we do statistical analysis of our data. So is there a statistically significant correlation between bacon consumption and change in total blood cholesterol level? We would have to run statistical analysis to know that. We are not doing that this semester in this class. In this class, we're just going to look at our numbers, look at the trends in our data, and we're going to make conclusions based on that. I know that's not 100% scientific, but the math prerequisite for this course doesn't require you to know how to do statistical analysis, and we're not going to teach that in this class. Okay, then based on that statistical analysis of the data, the scientists would discuss their results and make conclusions. And you're going to be doing that in this class as well. And ultimately, you're going to answer the question, did your data support your hypothesis? And hopefully the answer is not always yes, because some of the best discoveries in science are made when the data doesn't support the hypothesis. That's far more interesting, in my opinion, because then it's like, well, why? Why did it turn out this way? Was it my experimental design? Was my hypothesis flawed? Some of the best discoveries in science are when we get an unexpected result. And then ultimately, every scientist wants to publish their results in a peer-reviewed journal. Sorry, that news popped up there. We want that peer-reviewed journal publication so that there's oversight and scrutiny of the research, so someone else can retest our experiment to see if they get the same results, so they can build on previous knowledge. We didn't get to where we are today by people keeping their results under wraps. We want to share knowledge so we can build on that knowledge. We want to find cures for diseases. We want to solve all these big problems in science today by sharing that knowledge. And we'll talk a little bit more about why that's problematic throughout this semester, because with biotech and private industry running research, a lot of times they don't want to share their knowledge, and cures don't get made because nobody wants to share their information. If there's millions of dollars at stake in developing a drug, and they want to hide their results until they're the ones who discover the drug. So sometimes progress is slow when information isn't shared, but it just really, again, emphasizes the importance of publication and scientists sharing their information. Obviously, this semester, you're not publishing your results in a peer-reviewed journal, but we're still going to follow all of these great steps of the scientific method in everything we do. Okay, that is it for the scientific method. Again, there will be a separate video on how to create tables and graphs. Thanks.